Inside every living cell, mitochondria produce the energy needed for life. Their inner membrane contains the electron transport chain, the final stage of aerobic respiration. The folded cristae provide a large surface for protein complexes, allowing efficient energy production through oxidative phosphorylation. NADH and FADH2 deliver high-energy electrons from glycolysis in the citric acid cycle to begin the electron transport chain. At complex 1, NADH releases electrons. Their energy pumps hydrogen ions into the inner membrane space, creating a proton gradient. FADH2 transfers electrons to complex 2. This complex does not pump hydrogen ions, so it contributes less energy for ATP production. Coenzyme Q carries electrons from both complexes toward complex 3. The continuous electron flow prepares the mitochondrion for efficient ATP synthesis. At complex 3, electrons arrive from coenzyme Q. Their energy pumps more hydrogen ions across the inner membrane, strengthening the proton gradient needed for ATP production. Cytochrome C receives the electrons from complex 3. This small mobile protein rapidly carries them across the membrane surface to complex 4. At complex 4, oxygen acts as the final electron acceptor. Oxygen combines with electrons and hydrogen ions to form water, completing the electron transport pathway. As hydrogen ions accumulate outside the inner membrane, a strong proton gradient is created. This stored energy becomes the driving force for ATP synthesis. Hydrogen ions now flow back into the mitochondrial matrix through ATP synthase. The movement of these protons powers the enzyme like a microscopic turbine. ATP synthase converts ADP, an inorganic phosphate, into ATP. This remarkable molecular machine.
Because NADH donates electrons at complex I, it drives greater proton pumping and produces approximately 2.5 and generates about 1.5 ATP molecule. Electrons move sequentially through 4 protein complexes. Proton pumping and ATP synthase converts this stored energy into ATP. Certain toxins and inhibitors block specific components of the electron transport chain. When electron flow is interrupted, proton pumping stops, asynthesis declines dramatically. Uncouplers allow hydrogen ions directly across the inner membrane.
Efficient oxidative phosphorylation is essential for organs with high energy demands, including the brain, heart, and skeletal muscles. Defects in mitochondrial function.
The electron transport chain is the final stage of aerobic cellular respiration. Through coordinated electron transfer, proton pumping, chemiosmosis,